So you're going to have the study guide, you're going to have the Pearson, and you're going to have the other practice problems in here, right? So it's going to be a pretty eventful weekend for AP Bio so that you can take your unit test next week. Yes. Don't worry about unit card. So, they don't worry about it.
Oh, sorry, no, I was trying to figure out what work we did yesterday.
What work we did yesterday? 56 to 60.
Oh, okay.
Yeah. Well, that's what you showed me, right?
No, but Michael said that there was homework.
I didn't assign homework last night.
Oh, I thought it was like the practice problems that you're supposed to do?
No, not the next set yet.
Oh,
Did
okay.
Did I?
Because there was an FRQ in the back, but he said we didn't have to do it. So I don't know if we were supposed to
Oh,
do it.
yeah, because we didn't get through all of this yet to...
Oh, but it's after we get up?
Yeah,
Oh.
yeah. All right. So, so we said yesterday our phosphate group gets added to the 5' side of the ribose. Yes. Mm-hmm. And we're like, oh, this is the 5', right? So right here, where this oxygen is, we're going to name that 1, 2, 3. Right here with this hydrogen is the 3rd carbon. Number 4 is right here. 5 is right there. So the 5'. What page are we on? We are... right here. So again, we have a 5 carbon sugar, right? A pentose. The 5' is where the phosphate group is attached. So we're all squared away with that. And that's on our DNA molecule, right? Or our nucleic acid, right? Here's the nucleoside, the portion without the phosphate. And then the phosphate group, and then our sugar. Remember ribose and deoxyribose? What's the difference between ribose and deoxyribose? It's missing the oxygen, right? So deoxy, right? It's deoxygenated, I guess you'd say. Cool. Polynucleotides. Poly meaning many, nucleotides, right? And here we have, what is this here?
A pentagon.
Okay, it's a pentagon. It is not oxygen. What are these blue ones? They're pentagons, but they are what? It's either the ribose or the deoxyribose, right? Either one. We don't know right now. But yes, it's our ribose or our deoxyribose, right? It depends on if we're just leaving out this other side, or if this is a single strand, this is our RNA. If it's only half of the double strand, it's our DNA. Right on? These little yellow circles, what are those?
R.
What's that? Say it again. Not the R group is in the amino acids. Right? So if we go back to amino acids.
Right here.
Yeah, that's the R group, right? But now we're looking at a whole structure. So I'm telling you guys, polynucleotides. We just looked at a nucleotide right here. Right? Look at this. This is adenine. What does this yellow circle represent then? The phosphate groups. Absolutely. So we've got our pentose right here. We've got our phosphate group. That makes this the... What is the orange things? The nitrogenous base. Absolutely, right? So this, how many sides? 6. So the single ring, 6-sided nucleic bases are what? Shh. Tristan. The 6-sided single ring Nucleic bases are what?
Pyrimidines.
Close. Pyrimidines.
Pyrimidines.
Cool. The double ringed, one pentagon and one hexagon, are the purines. So this could be adenine, right? Adenine is a purine, right? So here we go. Phosphate groups link adjacent nucleotides.
Sorry.
It is directional linkage. 5' to 3'. Where is the phosphate attached? On the last slide, where do we say phosphates are attached? Remember when I said it's important to understand where the phosphate group is attached? It is attached to the 5'. Then you can orientate this nucleic acid. This is the phosphate attached right here to the 5'. Right? Yes, no?
Yes.
Cool. That's 1, 2, 3, 4, 5. Right? So now I know what direction to read the DNA or the RNA in. Because I know the phosphate is attached to the 5'. There's our nitrogenous base, our sugar phosphate backbone. There's the 5' phosphate head and the 3' phosphate tail, or hydroxyl tail. Any questions about this? Remember yesterday when we talked about proteins, right? And it being directional, you had the amino N terminus and the hydroxyl terminus, right? When we link amino acids together to make proteins. Does that sound somewhat familiar from yesterday? Please? And this derives from the DNA or RNA being read in the proper order, because that gives us our amino acid order, and that so the order of the amino acids we have to have amino here, and then everything starts building amino to that hydroxyl. Then there's hydroxyl here, so amino joins there. So is there hydroxyl here? Amino joins to there. Hydroxyl here, right? And that's where we get that chain. The sequence of the bases along the DNA or mRNA is unique for each gene. The sequence of bases. What were the bases again? There's 4 of them. Well, when we talk about DNA, there's 4. G, T, A, and C, or A, T, C, and G, however you want to, right? And then if we're talking about RNA, the difference is what? Uracil as opposed to thymine, right? This tells us what amino acid sequence is going to come in. It's read in 3-letter groups. Do you remember what we called them yesterday? Look, we what did you say? What? So codon is part of it. Codon is what is brought in. Or oh yes, you're right, codon. I'm thinking ahead, dude. The codon, right, is our 3-letter group, and then the anticodon comes in, and that's what brings the amino acid, right? So this is the codon. I don't know why I'm thinking ahead. I'm sorry. So this is our codon, right? Our 3-base group that tells us what amino acid to bring in. The anticodon is complementary to the codon, and that's what brings in the amino acid. Are we good? Questions? No? No questions? Everybody understands this 100%? So we can give you a quiz right now and you'll be like, I got it.
That's because I was yesterday, so I don't.
Right? You miss a lot, dude. And remember we have our 4 structures of protein. The amino acid sequence describes or determines the primary structure. From primary structure, we go to... Huh? Say it loud. What? After the primary structure of a protein, what is the next structure? Secondary structure. From secondary, we go to tertiary, which is the 3-dimensional functioning protein, right? 2 or more proteins come together to make what structure? Quaternary, yes, absolutely, and that's our functioning enzymes. Right on? Yeah. So the primary dictates the 3-dimensional structure of the protein, which is the tertiary structure. So if this pyrimidine becomes a purine by accident, then that gives us a different codon, which will bring in a different anticodon, which could bring in a different amino acid. If it brings in a different amino acid, it means the R chain or the characteristic of that amino acid may not be what it's supposed to be, so we don't have the correct primary structure, right? Just that bead of amino acids. If we don't have the right primary structure, could that affect the secondary structure, the folding of the secondary structure? Yeah, for sure, right? Where hydrogen bonds and stuff are supposed to go may not go now, right? And if that's messed up, could that mess up the 3-dimensional protein, the tertiary structure? Absolutely. And if you mess up the tertiary structure, this is where we get diseases and disorders, right? And especially if that tertiary structure is supposed to go into a quaternary structure and become an enzyme, then it's even more detrimental probably, right?
Yeah.
All right, DNA. So we have our double stranded DNA. On the other side of A is always T, and the other side of G is always C. It's just that simple. There was a scientist, Chargaff. We have Chargaff's rule, which helped us with this in the sense that the percent of DNA that is cytosine is equal to or about equal to the percent of the DNA that is guanine, and also the percent of thymine is also equal to the percent of adenine. What we notice, if you look at this, do 2 purines join together?
Yes.
Do 2 pyrimidines join together? No. It's always a purine with a pyrimidine, right? We didn't get this for a while. When we were able to X-ray DNA and able to see it, we were able to measure the distance from the outside of one piece of DNA to the other outside. And we had a measurement. Right? We can't see these guys. Right? Like, we can't look at and be like, oh, there's the guanine. Right? But what we can do is we know the width of the DNA. And we know the width of each purine and of each pyrimidine. And when we tried to figure out like how DNA is structured and right, we knew the width of it. Well, if you put a purine with a purine, are the purines the single strand or the single circle, or are they the double? Look in your paper.
What?
Adenine. Is adenine a purine or a pyrimidine? Don't guess. Adenine's a purine, right? And it's got the double rings. When we put two purines together, the DNA is too wide. When we put two pyrimidines together, the DNA is too narrow. The only way the DNA is the right size is when we hook a purine to a pyrimidine, right? This is how we started our structure for DNA, knowing, like, okay, what goes with what. We knew these had to match up with those. And that's it, right? And then we can start chemically marking them. Michelle, where you at? Edie? I don't know why I'm walking over there. Ashley? Where you at, Ashley? Ashley. Fiorella? Victoria? Where'd Victoria go? I saw her walk in. We're going to put this right here. Sabah? Not here, right? Sabah? Noor? And Clay's not here either. Thank you, sir. You too, buddy. All right. So, DNA structure consisting of two polynucleotides forms the double helix. Strands are anti-parallel. What does parallel mean? What does parallel mean? Yeah, they're going together, right? Not running into each other. What does anti-parallel mean? Well, so you would think, okay, anti-parallel, that should mean perpendicular, right? If it's not parallel, it should be perfect. That's not what it means, right? So, again, you've got your two strands. Right on? Where's my... This is what? What is the yellow right here? How many sides are on there? 5. What is it we've been talking about that has 5 sides in DNA and RNA? The what? Not the phosphate group, the... I know, right? DNA is made of 3 things, and we've mentioned 2 of them, and I said no. What's the third one?
So it's going to be the carbon. Oh my god, the deoxyribose stuff.
It's the sugar, right? So if this is the sugar, and this is my phosphate, right? That means this right here is what? Where's that phosphate attached to on the sugar? Where does the phosphate group attach on the sugar? On the 5', right? This is how we orientate the DNA then. We're like, oh, if this is where the phosphate is, right? Here's our inorganic phosphate or whatnot, or we'll just leave it organic phosphate. This is our 5'. We know it's attached to the ribose or deoxyribose, right? And then here's like an A. Cool, that's what we're showing right there. There's adenine. And then we know A pairs with T. So that makes this a T, right? And then we have our deoxyribose again. And then what is attached on the outside of that deoxyribose? It's right there. Well, what's attached to that? There's the deoxyribose. What's attached to it right there? Not where's it attached, what is attached? What's attached right here? Yeah, that's our phosphate group again, right? So we have another phosphate right here, right? Where is this phosphate going to be attached onto this deoxyribose? Now you can say it, yes, to the, not the other one. It's attached to the 5'. So if the 5' is here, the 3' is here, and this is where we make our little branches. Here's a 5' to 1, and then the 3', and then the 5', and the 3', right? And then this is going to follow that same pattern, which means this is what, I'm sorry, this is what part of the ribose, of the deoxyribose. That little arrow pointing out is the 3'. So that this runs 3', 5', right? That 5' there. Then you have a 3' here. Then you got a 5' here. Then you got a 3' here. Then you got a 5' here. This is anti-parallel. What I have on this side, I have its opposite on the other side. So when we say that it's anti-parallel, this is exactly what we mean. So that the very end of this, how we're going to tell you to think about it, is that this ends at 3' and this ends at 5'. Because this side has 5' up there, it's going to end in 3'. Because this has 3' up there, it's going to end in 5'. Stop. Do you guys get this?
Yes.
Cool. What's the anti-parallel string to that? Can't have those conversations where we're describing this stuff, dude. You're gonna miss a lot.
No. Oh, it sounds like
We should. What is that? Anyone? No, I wrote it up there. Really, go ahead, answer it. What's an anti-parallel string to this?
right the opposite. Or no?
Yep.
Okay.
Yeah. Well, give it a shot. 3. What is that? YSV. All right, so we got 5' and then it goes A, so it should just start with 2. Cool. So we shouldn't have that C on there. And then this one here should end in a G, right? And that should end in a G.
Am I missing something? How am I supposed to know what to do?
You just guess. That's what I'm doing. I just guess. No. So you've got this, right? And if I have this, what is the anti-parallel strand going to look like? Remember, anti-parallel is opposite. So we got 5'. So that makes this 3'. 3' makes this 5'. On the opposite side of an A is going to be a T. Opposite side of a T is an A. A, T, G, T, C, T, A, G. It's just the opposites for each of these.
Does this list opposite here?
Yeah. C, G, A, T, C. And here we have 5', 3', 3', 5'. So that's anti-parallel. Does that make sense? Yes, no? Okay. All right. So it should start with what? 3'. Because we have 5' here, we should have 3'. And then we just do what? Yeah, we put the complementary base pair, right? So with the T, or with an A goes a T. There. That's it. Don't make it that difficult. Cool? Yes, no, maybe so. All right. Held together by the hydrogen bonds between the bases. Right in here is the hydrogen bonding, right? Or right in here is the hydrogen bonding. Here we have the anti-parallel ends 5 prime, 3 prime. C binds to G, A binds to T. That's it. But this should all be, the only thing that should be new is the term maybe anti-parallel, right? From 9th grade, you should have all this, yes? The C's, the G's, the A's, and the T's. Yes, no? We're just throwing a new term in there, anti-parallel, the 3 prime, 5 prime. Are we good? Move on. Oh, yeah. And now there's another little practice at the end of that page that you can fill in. It says you're given a little bitty strand. What is the complementary strand?
So,
Okay, so you're good there.
Yeah.
And then what goes opposite of cytosine?
would it be the T?
So always, right?
Okay.
A's and T's are always across from each other.
Okay.
C's and G's are always across from each other. So if I have a cytosine here, I have to have a
The T?
no. A's and T's. Look. A's and T's always are opposite each other.
Oh, so it's C.
C's and G's are always opposite each other.
Oh, okay.
Here's a C, so what's going to be here?
A G?
A G.
Oh, okay.
That's it. Yeah, just make it that simple, Tristan. Right on?
Okay, yeah.
What's up?
This is right.
I don't know. I make this up. G, T, A, C. Yeah. Are we all good?
It's anti-parallel again, right?
Mhm. Yeah, so down here it should be 3 prime, G, T, A, C, A, G, T, T, G, 5 prime. Cool? Right on. Good.
I can write what's
This is what carbon it's attached to. That's all that means. And so it's always 5 prime, that, and then 3 prime.
so when they say carbon, I just switch these two.
Mm-hmm. Mm-hmm. That's it. All right, RNA. Single-stranded polynucleotide, not double-stranded. Variable in shape, sure. Due to the base pairing within the RNA, we're not going to get deep into that. Adenine binds to uracil. It's single-stranded. How does adenine bind to uracil? Are these guys binding to each other right here? No. So if we have our 5 prime on our DNA, right, and we've got A, T, T, A, C, C, G, A, T, A, and our 3 prime, right? Then here, we're going to be this. Right? And here's our DNA. When we do transcription translation, this is going to make more sense. What did we say about the DNA? How often do we want it to leave your nucleus? Never. It will be destroyed. If your DNA is destroyed, that tends to be bad, right? What leaves the nucleus instead? RNA. So remember we talked about the DNA had to be rewritten, right? Transcribed. So what has to happen is the hydrogen bonds in here have to be broken, right? And we'll get into this. We call it unzipping. Yes?
The hydrogen bonds are what connected?
Yeah, it's connecting the A's and the T's and the C's and the G's, right? So what has to happen is these bonds have to be broken It's going to drive the DNA crazy because it really wants to be bonded. This thymine really wants to be attached to that adenine. But we put a little blocker on there so they can't stick to each other, right? And what comes in is a little piece of RNA. Across from A is going to be what? It's going to be what? U. Uracil. This is RNA. Uracil. Right? And then adenine, uracil, guanine, cytosine, uracil, adenine, uracil. And then a 5 prime. This is our RNA. So your body sends a signal, says, hey, I need this gene made because I need this protein. And your body says, okay, untwist this segment of DNA, right? Open it up, unzip it, write this RNA. This is going to take off, right? These guys are going to, this DNA is going to go back together. So after the RNA comes out, oh yeah, these bonds come back and our DNA stays intact. Cool? And now this little RNA segment cruises out, like we said yesterday, goes to the ribosome and gets translated. And this is where the amino acids get brought in. Cool? So when we say adenine bonds to uracil, we're talking about in the transcription and in the translation. Because remember, I said in the ribosome, right, the RNA is going to come in and it's going to be like GAC on that RNA. Something has to be able to attach to it in here. What's that going to be? CUG. And this is going to have the amino acid on it. That's what's going to come in here. We're going to weld them together with the peptide bonds that we talked about yesterday. When we get the end signal, that's where that primary structure is going to take off from the ribosome. It's going to start to fold into the secondary structure, go to a tertiary structure, possibly quaternary. Cool? So hopefully this helps tie I'm sorry, I wrote on. This helps tie together
That's what I need, so.
what we started talking about yesterday. So cytosine bonds to the guanine, right? We have practice starting on page 70, and it goes through to page 75. So we've got 70 to 75. And then if you back up to page 66, now we can write your practice FRQ. So please write yourself a note. Practice FRQ on page 66. Practice page 70 to 75.
What?
Yes?
Is this not notes?
What?
Is this not notes?
How did I miss that one? Where's it at? What page?
That's 72.
Oh, did I miss? Sorry. 71. Oh, yeah, lipids. My fault. All right, don't write that note yet. I'm ahead of myself. Where'd my clicker go? Who stole it?
You placed it right here.
Why is it always you, Scarlett?
What?
All right, so we'll hit the practice tonight. I'm sorry. Lipids. Totally forgot we didn't do lipids yet. Lipids. Did I take your packet? Oh, I'm bringing it over here? That's kind of funny. Lipids, right? We said we're fats, waxes. I'm missing one. Oils. Man. Do not include true polymers. Polymers are made up of what? Monomers, right? So what does that tell me? That tells me that a lipid is just a lipid. It's not made up of a bunch of little things. Did you break your necklace?
No.
No, I need to tie it back on.
Oh.
It's like a little bit.
Sure, generally small in size. That's relative, though, to what you're talking about, right? Compared to an atom, probably not that small in size. Compared to the Earth, yeah. Often not considered to be a macromolecule. Non-polar. Again, I don't know how many times we mentioned this. If it is non-polar, it doesn't have the charges, so it's hydrophobic. I think we mentioned this the other day, though, right? The types of lipids, the fats, steroids, and phospholipids. God bless you, my job. Carbon, hydrogen, glycerol, fatty acid. Okay, cool. All right, move on real quick. Yes? No? Maybe so? Yes. All right. Fats composed of glycerol and fatty acids. We mentioned glycerol the other day. What did we say it is? Any ideas? Any remember? What did we say glycerol was? Has to do with storage, right? It's the form we, yeah. Classified as an alcohol, because it has a hydroxyl group. Fatty acids, long carbon chains. Carboxyl group at one end. Okay, big deal. That's not terribly important. 3 fatty acids join to a glycerol. Ester linkage. You don't need to know what an ester linkage is for the purposes of our test or the AP exam. So don't like, oh my gosh, what's an ester linkage? Don't worry about it. No. This you will, though. Our fats we can classify as a saturated fatty acid or an unsaturated fatty acid. And it is going to have to deal with how the hydrogens are arranged around the carbons. Saturated fatty acids. There is no double bonding between carbons in the carbon chain. So we get more hydrogens. If something is saturated, what does that mean? Have you ever heard of the word saturation? What does it mean? What?
Be put into.
Kind of to be put into. Almost satiating, kind of. I think you could.
Oh, isn't it like more filling?
More filled,
Yeah.
right? If a sponge is saturated with water, it can't hold any more water, right? If anything is saturated, it is filled to capacity. It cannot hold anymore. So when we say we have a saturated fat, it is a chain of single bonded carbons where you can't put anything else in there. It is totally saturated. It means the same thing. It means it's a string of carbons that are surrounded by hydrogens. We'll get to it. Saturated fats or saturated fatty acids have characteristics. Generally solid at room temperature. What saturated fats or what fats do you know that are solid at room temperature? Butter. Sure.
Yogurt.
Yogurt's no. What? Yogurt. Oil. Oil is solid at room temperature.
Oh, never mind.
Lard. right? Like mayonnaise is not. No, that's not. Mayonnaise is an emulsification.
What does that even mean?
Don't you worry about it, dude. So things like lard, butter, right? Shortening, right? Half and half is a milk. Would you stop it? Unsaturated fatty acids contains one or more double bonds. The double bond is between the carbon, which means so I've got carbon, 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 carbon. Carbon, 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 carbon. Yeah, we'll go there. We'll go here. I don't know. All these carbons have their 4 bonds, right? Wait a minute. All these carbons have 4 bonds, correct?
Doesn't that one have 5?
Which one? Did I put 5? This one? 1, 2, 3. Yes. All right. They all have their 4 bonds, right? We said they can make, earlier we said carbon can make 4 covalent bonds, correct? And man, you can't see that back there. You guys in the back can't see that, can you?
Okay.
You can? We'll share the information with Oakley. So the top one or the bottom one, which one has room for another hydrogen? Do either of these have room for another hydrogen? The top one or the bottom one? Where?
Oh wait, no.
It's kind of a trick question, dude. If this double bond gets broke, it has room for carbon or for hydrogens. But with the double bond there, it doesn't. So we say it does have room. This bond has to be broken, and we can put hydrogens in there, right? Anyway, I drew it wrong, but right, but wrong. It's not scientifically right, but the idea is right. So that is what we mean by unsaturated. There is room for hydrogen if we break a bond. These fatty acids, unsaturated fatty acids, tend to be liquid at room temperature. What are some fats or whatnot that you know of that are liquid at room temperature? What kind of oil?
Avocado oil.
Okay, avocado oil.
Mayonnaise.
Again, an emulsification, man.
What even is that?
It's where you mix the things together, dude.
Oh.
But mayonnaise is egg. I just forgot. No, it's not mustard.
Oh. So we're eating raw eggs?
There's oil, egg. I can't remember what else is in there.
Wait, so we're eating
Yeah,
raw
it
eggs?
might be milk. Yeah. No, I don't know. They're probably pasteurized. Anyway, avocado oil, canola oil, sunflower oil, olive oil, huh? Vegetable oil. All of those things have what in common?
Oil.
They're made of fruit and vegetables.
They're all what?
They're made of fruits and vegetables.
Okay, and we call those things.
Phospholipids.
Fatty acids.
Oh, you're killing me. All fruits and vegetables are types of. Yes. Holy smokes. Yes. Unsaturated fatty acids come from plants. Saturated fatty acids come from animals, for the most part. Coconut oil is weird.
Wait, what about the spray on oil?
It's usually like a canola oil.
What's canola?
Canola is a plant.
It is?
Oh yeah, have you ever heard of anybody hunting canolas?
Maybe.
No, because it's not an animal, dude. We don't hunt canolas.
I mean,
So not to be confused with a cannoli.
oh,
Oh,
good.
cannoli oil. All right. Where does all this matter? This matters in the structure of organisms. Oh, wait a minute. Somebody mentioned butter.
That was
What's another term or idea you associate with butter?
Toast.
mantequilla.
Is there more than one? How do I want to say it? What? Not clarified butter, no.
Unsalted.
So we have not the unsalted and salted butter. That's this difference in baking. There's baking, yes. Because if you put salt in your recipe, then you don't want salted butter, dude, because it oversalts the recipe. Don't you know anything, Scarlett?
What? Okay, continue on.
All right, so we have butter and we have. Okay. Oh yeah. What is that? Margarine. Why do we have two different names for the same thing?
Because one's for cooking.
Nope. They both go on bread. You can use them the same way. One, you want to use butter for baking, but not margarine. Ooh. Why? Don't they kind of look the same, right? Country Crock and Land's End. Is it Land's End? Anyway, no, Land's End's a clothing company. Land O'Lakes. Yeah. Shh. Stop making fun of me. You know I'm sensitive. I'm sensitive. They're making fun of me.
Like the sugar and then Splenda?
No.
Is that what you're trying to say?
No. Butter versus margarine. Margarine is from plants.
Oh, that's right. Yeah, country crock is plants, right?
Well, sure. But how do we get? Well, wait. Is margarine liquid at room temperature? No. But I'm telling you that the fats from plants are liquid at room temperature. Why isn't margarine liquid at room temperature then?
Because it's lard. I mean,
No.
whatever you call it, like the mixture of stuff.
No. Well, yes, ma'am. It's what? It's saturated. Well, no, but it's unsaturated. But it's saturated. How? What do?
We build it.
Yeah. What we do is we manipulate the unsaturated fatty acid and we force a hydrogen in there. And we saturate it. So we've taken what should be a liquid at room temperature and we make it a solid at room temperature. That is what margarine is. This is why you don't use margarine in baking, because when that bond is broke, it's not going to be butter anymore. It's going to act as a liquid. It's going to be more soupy in your baking mixture, so you use butter instead of margarine. Are you going to question me, Jamaica? This is where your cookies will fall and they won't rise as much too and stuff. But
Okay.
all right. What you know I used to be a chef, dude.
What? How would I remember that?
I've told you guys before I used to be a chef, right? Yeah. See? Choo. Side eye me, child. All right. Phospholipids. Where do you hear the term phospholipid?
Oh, I know this.
Phosphates.
It's in
If you were in my 9th grade class, you heard semi-permeable phospholipid bilayer, and that described what? That described the semi-permeable phospholipid bilayer.
So it's a layer that's not always there.
It's there. This is what we refer to for your cell membranes.
Oh yeah.
Oh yeah, now you remember? Yeah. It's a major component of your cell membrane. Semi-permeable phospholipid bilayer. Hydrophilic head. Hydrophobic tail. This is our lipid. This is our phospho, right? It's a phosphate glycerol up here for the head. Hydrophilic. It's loving water. This is going to be exposed to the aqueous side. Both the inside and the outside of your cell is going to be aqueous. Oh man. All right, so Yeah, start the study guide you'll get tomorrow. Start the practice problems in your packet, and please start mastering biology. Mastering biology is projected to take you 2 hours. So please get started on that. And have a good day.
Just keep it here.
How do I turn it off?
Just hold it. And like the blue light should stop flashing.